Hello, and in today's session I'm going to be talking to you about the basic monomers of proteins. We're going to be talking about amino acids. I'm going to just first of all introduce you to the functions of proteins in living organisms. Then I'm going to talk to you about the generalized structure of amino acids, and then how those amino acids can join together to form dipeptides. So I always like to say that you are really just a big bag of protein, and the bag is made of protein. Pretty much everything in your body is made of protein. Think about it. Your skin is made of protein. The collagen that holds your skin onto your muscle, made of protein. Your muscle is made of protein. Your bone, your cartilage, your tendons, all made of protein. Your hair, keratin, your nails, made of protein. Within your body, your internal organs are all made of protein. Your smooth muscle of your kidneys and your liver and your small intestine, the cardiac muscle of your heart. So these are all examples of structural proteins. They build up the parts of your body. The other category of protein within your body are the functional proteins, the proteins that do jobs. So, you're a big bag of enzymes. You are just a whole load of metabolic reactions occurring, and those metabolic reactions are catalyzed by enzymes. Think about how you digest your food, or how you build up new muscle or new skin, or how you make connections in your brain. All these things are catalyzed by enzymes: amylase, pepsin, catalase, and over 10,000 other enzymes in your body, all made of protein. Proteins are involved in transporting things around your body, like the obvious things, like hemoglobin is a big old protein that transports oxygen around your body that you're probably all familiar with. But in addition to that, we transport things in and out of our cells. So we have protein pumps and protein channels embedded in our cell membranes that enable molecules to be moved across. You'll learn loads more about different protein channels and protein pumps as you carry on doing the biology A level. And then hormones. You already know about loads of hormones, but almost all of your hormones are protein-based. Insulin, glucagon, serotonin, thyroxine. These are all protein-based hormones. A small proportion of your hormones, like estrogen and progesterone and testosterone, are lipid-based, but most of them are protein-based. And then think about all the receptors in your body. So think about how we are able to see color. The molecules rhodopsin and which is found in our rods and enables us to see black and shades of grey, and iodopsin, which is found in our cones and enables us to see all the colours that we see. These are protein-based molecules. And then if we think about our immune system, antibodies, these are big old Y-shaped proteins. We need proteins for blood clotting. So when you form blood clots internally or when you form scabs, these are proteins. What about the proteins that enable you to move your limbs? So we need to lubricate our joints so that we don't get friction that rubs the bones together and damages our bones. So we have this sort of little talked about fluid in our body called synovial fluid. Synovial fluid runs around all of our joints and reduces the friction as we move our elbows or as we move our wrists or our knees. These are glycoproteins, and these are super important to keep us flexible and healthy. It's also really worth talking about examples that are not human-based. It's really easy in biology to become really anthropocentric and really only focus on human or mammalian examples. But let's think about toxins that bacteria or other organisms might produce. These are often protein-based, like for instance the diphtheria toxin, which is released by the bacterium that causes diphtheria and causes necrosis of your internal organs, which is pretty brutal. And one you may have heard of, which is tetrodotoxin, which is often associated with pufferfish, but it's a pretty lethal toxin that's produced by quite a number of marine organisms. As a slight aside, if venom and toxins are a topic which interests you, I read this book a little while ago called Venomous by Christy Wilcox, which I would highly recommend if it's something you wanted to learn more about. One final function of proteins then, which you might find interesting, is as an antifreeze. A number of organisms that live at extreme cold temperatures are able to produce this protein in their bloodstream, which prevents them from freezing. Scientists have actually discovered this antifreeze protein in a wide number of organisms, including several species of fish, including the Greenland shark, snow fleas, and a couple of different species of frog, which is pretty incredible. Okay, so that's enough about the function of proteins. Let's now think about the structure. So proteins contain the elements carbon, hydrogen, oxygen, and nitrogen, and sometimes they also contain a sulfur atom. Proteins are made up of long chains of amino acids, and the property of any particular protein, whether it's a structural or a functional protein, or whether it's going to be an enzyme or a hormone or muscle, is determined by the sequence of amino acids. There are 20 different amino acids in nature, but all amino acids share the same basic generalized structure, and we're going to look at that together now. So here we see the generalized structure of an amino acid. Every amino acid has an amine group and a carboxyl group, as well as a centralized carbon and a hydrogen atom just here. What differentiates each amino acid is this bit up here, the variable region, which we donate here just as an R, but we would know that actually this area could be totally different depending on the amino acid. Now, don't worry about that. You don't need to know any of the structures of these variable regions. You just need to know this generalized structure, but you do need to know that these regions vary. And the fact that these regions vary give amino acids their different properties. So this variable region could make an amino acid polar or non-polar. It could make an amino acid acidic, negatively charged, or basic, positively charged. These properties of the amino acid will then determine how they will interact with other amino acids around them and the sorts of bonds that they will form and how they will behave within an aqueous solution. These things will then determine the structure and the properties of the protein that is then produced by this particular sequence of amino acids. As a slight aside, and this is not really important for your A level, you may have come across the term essential or non-essential amino acids and might not be sure what that means. Well, an essential amino acid, and there are 9 of them, are amino acids that you have to ingest, that your body is not capable of making. So the other amino acids, even if you don't eat them in your diet, you can still produce them through some metabolic pathway in your body. So as long as you have access to some amino acids, your body will just generate the others, the non-essential ones. But the essential amino acids, you have to ingest either through your normal diet or through supplements. You might have heard the term semi-essential amino acids. A semi-essential amino acid is essentially amino acids that might be essential in juveniles, in babies or in infants, but are actually not essential as adults. So that's the difference between essential and non-essential amino acids. Okay, so the next thing that we want to talk about is how amino acids join together to form dipeptides and then eventually polypeptide chains. It's really important that we can contextualize this so we understand where within the cell this is occurring. So we know that amino acids are joined together to form polypeptide chains and eventually proteins on ribosomes. And within most eukaryotic cells, ribosomes are attached to the rough endoplasmic reticulum. So when we're talking about this metabolic reaction, the peptide bond joining 2 amino acids together, this is occurring on ribosomes. And just to contextualize it even further, so you've got a really good understanding of why this is occurring or how this is occurring, we know that the sequence, the order of amino acids in a particular protein is determined by the sequence of bases in your genes. So we know that our DNA codes for proteins. And a particular gene is a sequence of DNA that codes for a particular protein. And when we say it codes for a particular protein, what we mean is it's coding for the sequence of amino acids that will make up that particular protein. So it's really important that we understand that when we're talking about proteins and amino acids, this all links to the ribosome, the rough endoplasmic reticulum, and fundamentally to our DNA. So let's look at how 2 amino acids become a dipeptide. So we can see here the bond between the C and the N is our peptide bond. And these bonds will be made between subsequent amino acids, and all of the R groups would point in the same direction. So when you're drawing this molecule out, or when you're looking at pictures of it, it's really important that you note that it always in the same direction. So whenever we were to look at a long polypeptide chain, it wouldn't matter how many amino acids made up that polypeptide chain. So we could have a chain of literally thousands and thousands of amino acids making up a polypeptide chain. On one end, we would always have the amine group, and on the other end, we would always have the carboxyl group. So we could refer to the different ends of a polypeptide chain as the amine and as the carboxyl ends. So when we think of a polypeptide chain like this, just a long strand of amino acids joined together by peptide bonds, this is known as the primary structure of amino acids. And that's everything that we're going to cover in this video. So we've looked at the general functions of proteins, the fact that they're both structural and functional. We've looked at the basic structure of amino acids, and we've talked about the importance of the variable region. We've also tried to put into context where amino acids would be joined together, as well as looking at how a peptide bond would be formed, both to form a dipeptide and a polypeptide chain. Just to check, I'm sure you already understand this, but this term di means two peptides joined together. And this prefix poly, just means many amino acids joined together. And these make up the primary structure. The next thing that you will learn about is the secondary, tertiary, and quaternary structure of proteins. But we'll leave it there for now. Well done.